From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on December 4th, 2023. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin of the Biology Department of the University of Puget Sound in Tacoma, Washington. Today is the 19th episode of Matters Microbial, and as always, I appreciate every viewer, every commenter, and every listener. Thank you all so very much. First, one of the most valuable members of my Wunderkammer. A few years ago, I met an artist who worked at a local cocktail lounge that my wife and I frequent on date night. Talk turned to tardigrades, as it so often does, and the young woman, Lindsay, made me a sculpture of a warrior tardigrade that glows in the dark, as seen here. She named it Z for tough. It's also the last week of classes here at the University of Puget Sound. I wanted to give each student something to bring them luck during the looming final exams. So, for my seminar in virology students, each Vironaut received a glow-in-the-dark 3D printed bacteriophage as seen here. Some of them will use them as earrings, others as necklaces, or just to keep them company during exams. For my microbiology students, each Micronaut received a 3D printed tardidoc, as seen here, to bring them luck. It is truly a privilege to work with each and every one of these students. Finally, in honor of our upcoming guest today, who shares my interest in skulls, please enjoy this image of a skull carved from fossilized bacteria, stromatolites, as seen here. Several episodes of Matters Microbial have discussed the microbiome. And as some of the videos referenced for those episodes suggest, when we eat, we are in essence also feeding our gut microbiome. There are whole industries that have sprung up revolving around this concept. And who better to tell us that it really is true that you are what you eat, or more accurately, perhaps what your microbes eat, than Dr. Suzanne Devkota of Cedars-Sinai Division of Gastroenterology and director of the Cedars-Sinai Human Microbiome Research Institute in Los Angeles, California. Welcome to our Quality Quorum, Suzanne. It's been a couple of years since we spoke, and I can't wait to catch up with you, what you and your coworkers are doing.
Thanks so much, Mark. It's a pleasure to be here.
Oh, it's so good to see you. And of course, right behind you, artistic work. Oh my goodness. I need to up my game. What can I say? Oh, well, it's very kind of you. So I think a good thing for our listeners and viewers is to give a little bit of a thumbnail sketch about your background, if you wouldn't mind.
Sure. Yeah, absolutely. So I'm a bit of an unconventional microbiology path. My background is actually in nutritional sciences, hence, I guess, apropos for the topic of this podcast. So I just had an interest in nutritional biochemistry and how nutrients affect the cells of our body. And I did my master's in that in nutritional sciences and my PhD in a program called molecular metabolism and nutrition at University of Chicago. I did my master's at University of Illinois. And then I, during my PhD, had a rotation project, as we all do rotation projects as a PhD student. And I had no intention of studying the microbiome or the gut. I was really interested in diabetes research, and I had a rotation to fill a gap in my rotation schedule. And my department chair said, "Why don't you go to this GI lab?" I said, "Okay, fine." It was 2007, right? So sort of this inflection point, as we talk about in the microbiome field, when sequencing suddenly became accessible to people for, microbiome analyses. And so I was like, what am I going to do in this gut lab? And so we set out to do a really simple project looking at whether different dietary fats could alter the gut microbiome, which is a really simple question, but hadn't been looked at the time. And so it ended up being really interesting, and I fell in love with it. And that's how I started learning about, the microbiome as a discipline. And I decided to not do any more rotations and just stuck in that lab and went, all in on the gut and the microbiome field, and have really watched the field change since almost the last 20 years now, almost. And it has evolved, and it's been a privilege to be a part of it. What I will say is sort of interesting is, as an undergrad, which I was also at the University of Illinois in Champaign, I had, microbiology classes from some of the luminaries in the field, Abigail Salyers and Carl Woese, and I had no clue at the time who I was in the presence of, but something in there clearly stuck in my head. And I went through the route of thinking I wanted to do cancer research and, neuroscience, but I ended up in microbiology. And I'd like to think that there's some seed planted from both of them that somehow reemerged, later on.
Oh, that is such a wonderful story. Surely you know how influential Abigail, the late Abigail Salyers, has been to me. Really changed my life in so many ways.
Mm-hmm.
But since you had her in class, you were well aware that she was a force of nature.
Yes.
And yes, I used to call her Hurricane Abigail,
Mm-hmm.
and she was something else.
Yeah.
Much, much missed. She used to call her students her peeps, and then she would bring those Easter peeps, speaking of bad nutrition, which I'll never
I don't
forget.
remember that. I don't remember that,
Yeah,
but
yeah.
that's great.
Carl Woese, story for another day, but what a remarkable person, such a thinker.
Sure.
Quite
Sure.
remarkable.
Yeah.
You've been very lucky, and that's wonderful to hear. So, would you mind reminding the audience a little bit about the microbiome? Because I'm afraid of something. A lot of times when I talk about microbiology, I think that PR is odd for the beginning micronaut, as I call them. So
Mm-hmm.
they'll think that every microbe is bad. I call those devil microbes. Or every microbe is good, and those are angel
Sure.
microbes. And
Mm-hmm.
then most just don't care. Those are meh microbes. So what I would say is that for some people talking about the microbiome, things get overstated. So to give some perspective, just a few words about that might be appreciated.
Well, sure. I mean, I think it's important to know that microbes are not just bacteria. They're in the microbiome. We have viruses, fungi, which are really cool. We have protozoa. We have, it's a very diverse ecosystem there. Microbes may be the most numerous in the gut, but they're not the only part of that composition. And so I implore people to spend time thinking about the other microorganisms that are part of our microbiome. But this is a co-evolved system, since we kind of walked on land and we started breathing air, and the microbes made that possible. And I think that it's a really beautiful story of coevolution, and I very much ascribe to the holobiont theory, where our microbiome and our host is one. Yes,
Mm-hmm.
two separate genomes, but they are a system that work together. Microbes do things that we know we have devolved and no longer can do, and so we need them. They need us just as much. And we are one unit, essentially, our microbes and us. And so that I think that concept has made it very attractive to assume the microbiome is the new cure for everything. And for many of the diseases that we have no cures for, someone has studied the microbiome in that area. And unfortunately, if you look hard enough, you can find an association with everything. And I think so. There's been some overstating about the role of the microbiome in disease causation and things like that. But I think there's no question that the microbiome affects our biology in good and bad ways. But our microbes are not born with original sin. They're not inherently bad. It is context. And it is, what we do to them and expose them to, the foods we eat, our sleeping patterns, our social patterns, our cohabitation patterns, all of that shapes our microbiome. And so we always describe them in our lab as biosensors of our external environment, of our outside world. If we look at our microbiome, we can see early signs of maybe things that are going on in the body. So that's more than a thumbnail, Mark. I'm sorry, but I get very passionate on this overstating question.
No. Seth Bordenstein is right now dancing around the room about the holobiont, that concept that you were discussing. It's one that I try to ascribe to as well. The whole idea of the microbiome reflecting different things is fascinating to me, because we really are one entity made up of many different parts. And when you consider how many organisms live in the gut, I believe it's 10 to the 11th per cubic centimeter. I don't want that job doing that counting, by the way, but it certainly is true. It's richer and more diverse than almost any kind of environment on the planet. And so it wouldn't surprise us that it has such a fundamental effect. In addition, the amount of the nervous tissue in the gut that surely is being interacted with is fascinating to me.
Absolutely. Yeah, absolutely. The nerves and hormones and immune system, it's very, yeah.
I just, like I say, I teach about this stuff and I get really over the top. That's why I call myself a microbial centrist, and people make fun of me, but I'm among friends here, so it's okay. Would you like to talk about some of your work in the lab?
Sure. Yeah, absolutely. So because of my background in nutrition, most of my work just in my graduate studies and postdoc was really focused on, how diet affects the gut microbiome. And it's always been, my work has always been focused on disease states, and particularly in chronic intestinal inflammation, like inflammatory bowel diseases. But my background is actually metabolism, so I have an interest in obesity and diabetes and the potential interactions with gut microbes and more systemic metabolic defects. But I would say, my lab is located within an inflammatory bowel diseases institute, and so that's been my primary disease focus, and also because it's quite easy for us to get human tissue specimens where I'm located at a hospital at Cedars-Sinai. And so we have great relationships with our colorectal surgeons who, when we want to study things like, tissue pathology and how the microbes change in different disease states, we're very fortunate to be able to get access to patients and their information, their histories and everything, and their tissues as well. So half my lab is divided in two ways. It's divided into those who study diet and the microbiome and those who study disease pathogenesis in the microbiome. And then within those two topic areas, there's the folks who study the host and the folks who study the microbes. So we have people who are physiologists and we have people who are microbiologists. And it's really fun to see people work together and, brainstorm thinking from these very different perspectives. We have been in the nutrition side very interested in things like intermittent fasting, and its effect on the gut microbiome. We're interested, as are many people, into different fibers and fiber fermentation by the colonic microbiome. We're particularly interested in their role in altering glucose metabolism, so butyrate and the effect on certain endocrine cells in the colon. And on the disease side, we've been studying something really weird and fascinating called creeping fat, which is fat that wraps around the gut in disease states, and microbes translocate out of the gut into fat tissue. So it's a great microbial ecology question of bugs who are adapted to one environment move into another environment and have developed an adaptive advantage in this new environment. And so that's been a really fun project for us as well.
I've only heard a little bit about the latter, but now I was unaware you were working on it, which is what Wow. That's amazing.
Yeah.
And it's funny. I tell people that, by the way, I'm not going to put you on the spot. Are there truly any sterile parts of the human
Uh,
body? I see.
yeah.
I know. I know.
Right.
We talk about it in class, and it's like I talk about, how you can tell. So this idea of a co-evolved population that can help us even digest our food is remarkable.
It's, that I mean, that's when we talk about microbes, and historically, even before the microbiome was a field, we always talked about microbes in that capacity in terms of their
Mhm.
ability to make B vitamins and vitamin K and ferment fibers, and but it was always talked about what they do for us. And it is definitely bidirectional, but that's been probably one of the most deeply studied aspects of host microbe coevolution is the nutrient and vitamin production by microbes.
Also, is it not true that some of the microbes in our gut are essentially farming us for mucus?
Yeah, I mean, yes, they do. Those are, very adaptive systems. Actually, in our intermittent fasting studies, one of the obvious bugs we would look at is Akkermansia muciniphila, which degrades mucus if other substrates are not available. And so when we fast, a mouse for 24 hours, they have access to water but no food. We see this bloom of Akkermansia and this degradation of the mucus layer. And then when we reintroduce food back, the Akkermansia is suppressed again and the mucus layer is restored. So it's kind of a beautiful system. It's not, I don't view it as harmful. It's just an adaptive system.
Well, I mean, this comes back to the idea, and it's something that I get my metaphorical little hammer. Don't say that. Don't say that. Don't say that. This whole idea about a perfect microbiome. It's like when someone gives me a choice between three things, and I say yes, because the fact is you can't really make a comment like that. The whole thing that I'm getting at is that it's a constantly moving series of interactions, and the time frame is what interests me, and that's why your intermittent fasting work fascinates me to some degree on this, because some of us are used to microbes that have like very, very extended time frames, like down at the
Mhm.
bottom of the Marianas Trench.
Mhm.
But as I tell the students, what did you have for breakfast? What did you have for lunch? What did you have for dinner? Think about how that looks to the microbes in your gut.
Mhm. Yeah, absolutely. I mean, they're not seeing the hamburger or the broccoli. They're seeing the chemical. They're seeing it's they're just seeing the currency of that food. They're not seeing, they're like, ooh, hamburger, I love that. And so it's in when you break it down into exchanges and things like that, you can start to think about diet microbe interactions in a more systematic way. But you're absolutely right. I mean, we in this particular study with the intermittent fasting, one of the lovely things you do when you're a trainee, I had to collect stool samples from mice every 6 hours for 24 hours, and from like many, many, many mice, which if you've done that is very difficult to do, to get them to do that often. And so, but what we found was the peak shift in the microbiome was at about 18 hours of fasting. So we saw the delta was the biggest in the shift. It was just an objective observation. And then when we reintroduced food at 24 hours, it just rebounded right back. And so the way it looked before the fast. So there is that surface level, like meal to meal shifts that everyone's microbiome will experience. Is it consequential or inconsequential? I don't think we really know, but we know it can. And so the question we were asking is, okay, what if you now do that often? And so a lot of people ask, how do I change my microbiome? They perceive it's not acting up or something, but how do I change my microbiome? It's got to be a daily activity. It's got to be a daily shift in your diet or a daily shift in something because that microbiome is so resilient, it will rebound. And so, diet is probably our best tool to do it. And but a single meal will shift it, but it won't fix that shift in time.
That's really interesting. Doesn't that lead us a little bit into And again, I'm not trying to stir up trouble or anything, but the whole business of the probiotic and prebiotic industry.
Mhm.
And I know
Yeah.
that most of our listeners and viewers have heard about it. So the idea is, can I introduce, quote, please forgive me, the proper microbes? See what I mean by angel microbes?
Mhm.
Or can I introduce the right kind of food for those microbes, the prebiotics?
Mhm.
What are your thoughts on this?
Yeah. I have a lot of strong feelings about this topic. So I used to be in the camp that says, after you take antibiotics, you should take a probiotic. It gives you the best chance at rebounding and having proper resilience and all that sort of stuff. But I'm always open to falsifying my beliefs and my hypotheses. And I now have seen data and evidence that suggests that A, um, taking that probiotic course after antibiotics can potentially delay normal recolonization of the gut, and can potentially be detrimental for certain populations. So, what I now recommend when people ask me that is really to increase the diversity of foods in your diet, especially of vegetable-based products, because Rob Knight's group from the American Gut Project showed nicely, I think, in the paper that the diversity across different populations around the world of the microbiome was attributed to the number of different kinds of plants they were consuming. And so I say, try to recolonize naturally. A diverse diet begets a diverse microbiome, and so do it that way. Um, but I think there are certain cases where a probiotic could be used for benefit. One maybe is if you've done an in-depth sequencing analysis of your microbiome, which is few people, but you're missing a key bifidobacterium or lactobacillus, then taking a probiotic to fill that niche could work. If you already have the bug, most of us already have many of these probiotic bacteria in our gut. If you already have it, that niche is occupied. If you take a probiotic, you'll just waste money and poop it out. And so it won't colonize because there's no available opening for it. But if that niche is open, then maybe it will work. The other population where data, and I'll stop my rant there, is the other population where it seems compelling that probiotics could be beneficial is in young children because their microbiome is still malleable, is still forming, and for babies that are born with things like necrotizing enterocolitis and conditions like that, there have been some compelling evidence that probiotics could be beneficial there.
That's amazing. I mean, so when a person goes to the store and they buy some probiotic from the dairy section, that's usually where you find them.
Mm-hmm.
I haven't seen the work. I'm sure it's been done. How few of those actually make it to your gut because they
Mm-hmm.
have to go through the stomach. And is it just a numbers game?
Mm. Yeah, it can be. I mean, I know encapsulation technologies have come a long way,
Hmm.
in food technology space. Um, and so, but if not, if it's yogurt, if you're just taking yogurt, that's a free form microbe, then it's a numbers game, because you're just hoping that some of it will get through. If it's encapsulated, I think the likelihood that it could get past certainly the stomach those products are better than they were today. So I'm a little less concerned about like encapsulated probiotics getting to the appropriate place. But food sources, yogurts, kombuchas, other fermented foods, I never recommend them as a probiotic food. I recommend them as a postbiotic food for all the metabolites because those
Hmm.
are the key functional unit and where you will likely get the benefit of a microbial food. So it's less about, I don't care as much about whether it colonizes or not. I care about the stuff in the gamish.
It's really interesting to me because I sometimes, and I know I'm being flippant when I say this, but the idea of, say, ingesting a microbe or encountering it in nature, I call them transients because they're not co-evolved.
Mhm.
So how do they manage to get a foothold? And we know what happens sometimes with, even though a lot of folks don't like the word, I think when it comes to Clostridium difficile, I can say dysbiosis, and nobody will be upset about that imbalance when you have that localized bloom. But for the most part, setting up shop essentially in the gut is something that seems kind of difficult because of the density of microbes that are there. Or am I missing something?
No, you're absolutely right. I mean, competitive exclusion, as we call it, is a key principle, in microbial ecology. And so, if you have a diverse microbiome, and diversity is often a key to intestinal health, but if you have a diverse microbiome, then many of those niches, homes in your gut where microbes like to live, it'll be like no vacancy. A bug comes through and there's no place for them to camp out.
Mm-hmm.
And but if you have taken recently antibiotics and you've now wiped out some of those homesteads, and now it's kind of like, hey, there's a lot of places for me to unpack my bags. And so keeping a diverse, robust microbiome can exclude a lot. I mean, we come in contact with quote unquote pathogens all the time. You go to the gym and touch the gym equipment, you come across, you go on the subway or you just go to any door handle or shaking someone's hand. I mean, this stuff is like all over all the time, and we're not getting sick all the time. And so there's some in the food we eat. There's stuff often on the foods we eat. So it's that competitive exclusion that really is quite effective, and we can thank our microbiome for that.
So, has anyone looked at the possible influx of microbes carried on the food that we're consuming? And secondarily, I've often heard people talk about a colorful diet, that is to say, lots of fruits and vegetables. Is that a good guideline for people to kind of assure a diversity of different types of fibers?
Yeah, good question. I mean, when I talk about diversity of the diet begetting a diverse microbiome, I'm really talking about it's actually the composition of the food, and not about the microbes on the food. But honestly, I don't think that we've ever really taken that aspect into account. So that may be a factor that sort of an additional, contributor to diversity. But it's really about diverse foods will have different fiber compositions, will have different micronutrients, macronutrient compositions, and different, microbes have specialized capabilities to break down certain foods. If we don't eat that food, that microbe will have nothing to do and no food to feed it, and it will die out. And so it's really about the composition of the food matrix itself. Um, and what is a healthy diet? I mean, a colorful diet is a good guideline. I think it really that really is regarding the vegetable component. Red fruit, red like peppers and blueberries and greens, and there aren't many colorful meats that we can eat. So I think it's really that component, which if I'm as I mentioned, earlier, that from the MBio study where they said the number of different plants that you eat, it was related to diversity. I think that sort of supports the colorful diet idea. So I think you could use that as a guideline. But really, what I would say is, and I'll use, the carnivore diet as an example. These are people who will eat only meat. That's it. No plant, no nothing, just meat, and often just red meat. And if you do that, you will support bugs that are really good digesters of just protein and just amino acids, and you will lose everything else. And so what happens if you suddenly decide to stop doing that and eat something else? You will not feel very good at all, because your bugs will be like, I don't know what to do with all this plant material. And so you never want to be caught in a situation where you travel or you change your diet and you don't have the right bugs to digest it. So um, it's like that concept of functional redundancy. You want many bugs that can do the same thing, so that if something happens, you still got someone to pick up the slack.
So we've covered a lot of things here that I think people are kind of interested in because, for good or for ill, there have been a lot of products that are thought to modify the microbiome that are sold over the counter now. And what I really appreciate is how you're saying it's really more of a nutritional aspect, which I'm going to hear about when I get home, because my family knows I could eat tacos for every meal. And so probably that's not a good idea. But I think the idea of having a kind of a lot of variation in diet to keep the diversity going seems to be a pretty good idea. What do you do?
I do really exactly that. I mean, I actively try to make sure there's diversity in my diet. I mean, um, there's a spectrum of I hear all of it. So I hear people who want to know about the carnivore diet and people who want to know about strict vegan diets and their microbiomes, and anything that's restricted is going to be restricting your microbiome. So but I'm an omnivore. I will eat meat, I will eat fish, I will eat plants. And I'm a big fan of fermented foods. I am, and I try to incorporate some fermented foods in my day, even though the data is still, we still don't have a lot of data on why they are beneficial, but stay tuned, there is data coming out on that. But I'm just a big fan of putting microbes in my body and seeing what happens. But because I do believe they're not all bad, as you mentioned, and so
No.
we and they're around us, and that's how we evolved, and we evolved in a microbial environment, and we still do, and that's probably the way our body likes it, is to be, we've only seen detrimental things happen when we sterilize ourselves too much.
Oh,
And so
absolutely. I mean,
yeah.
when we were hunter gatherers, I mean, I'm not suggesting that what happens is people say hunter gatherer diet, and then someone's trying to sell packaged meals, the hunter gatherer diet meals, right?
Right, yes.
I understand where that comes from, but still, the fact is, just being diverse in what you're doing is important. Do you
Exactly.
remember the story from a couple of years ago about the genes involved with breaking down the polymer in sushi seaweed, the nori,
Yes, yes, that was a fascinating
and,
study.
and how those genes, which originated from marine microbes, had been transferred by horizontal gene transfer into gut microbes in some populations, but not all.
Hmm.
And this is part and parcel of what we talked about before, this connection between the environment and us, even on the microbial level. It's
Hmm.
just an amazing idea.
Hmm. Yep, absolutely.
I think one thing we haven't talked about is the role of the microbiome, diet, and some forms of disease. And I know that you do some work in that.
Hmm. Yeah. I mean, it's still nebulous. There's a lot of things we don't know, in part because running human diet studies is very challenging. You rely on questionnaires, what did you eat today? And people, when they have a microscope put on them, are not always truthful about what they truly ate, or they alter their behavior so they look, so. Um, uh, so that's been a challenge, not just for microbiome diet studies, but diet studies, period, of all kinds. And so, we tend to, when we want to start on a new diet study, we tend to do it in mice, because they are very compliant. They will eat whatever you give them, and it's just more controlled. And then if we make an observation, then we might embark on doing a prospective human study, administering a certain diet to people. Um, but we've really been interested in how diet is more on the driving side of disease. And we're starting to think about how it can be used therapeutically to prevent disease. So you have to establish one before you approach the other. And we've been focused really on inflammatory bowel diseases, so chronic intestinal inflammation, which the gut is the highest density of microbes in the body. So studying intestinal inflammation is somewhat low-hanging fruit. And when we look at the foods we eat, we know we can directly modulate the gut. So it's a little bit more of a straightforward system. And we, it's it, honestly, the one most compelling thing that we know about diet is that the fiber content of the diet, and it's fiber so not sexy. It's probably the most boring of all of the nutrients and macronutrients that we consume, but it is the one truly that impacts the microbiome probably the most. And the microbes utilize it in really beautiful ways. I've sort of dived more into the fiber metabolism literature, and there's been people who've been studying this for a long, long time and doing great work in this space. But it's really the interactions are quite amazing. And, one pathway that I'll just highlight that I've become very fond of is um, microbes in the gut can metabolize fiber, and they often will produce short chain fatty acids, and one of them being butyrate. And um, enteroendocrine cells in the gut produce hormones that circulate to the pancreas and help regulate glucose and
Oh,
diabetes and so on. And so there's the butyrate can actually promote the production of GLP-1 and enteroendocrine cells, and that GLP-1 is the basis of drugs you hear about like Ozempic and Wegovy, which are the new hot weight loss drugs. But essentially, your microbes could theoretically do the job of those medications if you feed them appropriately. And so it's a whole new kind of paradigm thinking about microbes and metabolic diseases and by feeding them appropriately. So to your point, you are what you eat and what your microbes eat. I mean, it really is, you can affect your health by altering specific pathways. And I think by taking a really mechanistic approach, I think there's honestly something there. And so we're kind of exploring aspects of that. But up until more recently, we've been focused on how the lack of things in the diet or too much of things in the diet drive inflammation. And we were studying fat content, saturated fats, for example, and we moved a little bit away from that and are now studying a little bit more of the therapeutic angle.
it's really interesting stuff. Can you explain to the listener when you say fiber? I know what my mother meant when she said
Mhm.
fiber. What do you mean?
Well, this is one of the most, it's a great question because it is a very confusing question. Because if you look up fiber, you will see no two sources call the same fiber the same thing. So when I talk about fiber, I talk about fermentable versus non-fermentable
Got
fibers,
it.
meaning fibers bugs can do something with versus fiber that they can't do anything with, but are important to consume because they just provide bulk and keep things moving. And so I'm specifically talking about fermentable fibers, but a different definition, soluble and insoluble, are not the same thing. Those are not interchangeable terms. And so it's important, great question to qualify what you mean by the fiber and the way you're talking about it, because the definitions are not all the same.
So my fear is that someone will listen to some of this and go trundling off to the drugstore to look for fiber supplements.
Hmm.
And my knowledge of people who study this are more interested in dietary changes than supplements.
Hmm.
Would that be correct?
Yeah, but I mean, the supplements can do some of the job of dietary fibers. The one of the reasons why I have kind of steered away from some of the dietary fiber supplements is because in order to be palatable, they tend to have added sugars to them, and so I tend to, and surprisingly, like Metamucil has like quite a bit, but now maybe they're sugar-free versions. I'm not sure. So I always look at labels before I buy any sort of supplement, and I make sure there's no added stuff that kind of ultimately counteract the good thing I'm trying to do. So that's why diet is a safe bet. But supplements still have a role and can be fermented by microbes as well.
If I wanted to consume items that were high in fiber, what do I look for to make sure I'm getting sufficient fiber in my diet?
So, total fiber you can find on a label. So if you start there, you're starting off in a good place. And we all should be eating about 25 grams as a minimum per day. Most people eat 10, maybe. So just trying to up your fiber is is if you can hit your 25-gram goal, you're likely going to at least get half of that will be fermentable by your microbes. But there is no label that says, okay, the total fiber can be further broken down to fermentable versus non-fermentable. And I think there's even some research still being done on what is truly, there might be new fermentable fibers that we haven't really explored before. But there's a lot of products on the shelf these days, probiotic drinks and things like that, that actually have inulin-based fibers in them. If you ever see inulin on a label, that is fermentable. It can cause people a lot of bloating at the same time. And so if you're in that camp, inulin may not be right for you. But if you can handle inulin, that's a great one. Microbes love it. Um, Also, people have studied resistant starch, which is like green bananas, which don't taste very good, but green bananas are fantastic for your microbes. Those are some of the most kind of studied ones. But my general guideline would be try to hit that 25 grams of total fiber today, and you will support your microbiome in terms of fermentable fibers.
I've been reading in the literature about this idea of ultra-processed food as being a problem, and I'm wondering if anyone you know of has looked at the role of ultra-processed food on changes in the microbiome.
Yeah, this has been a very hot area over the last maybe almost 10 years. People have been studying this in artificial sweeteners has sort of been the most newsworthy area, showing that certain artificial sweeteners versus others can detrimentally affect the gut microbiome. There has been work from Rob Britton's group looking at dietary trehalose promoting C. diff blooms. Andy Gewertz's group has been studying food additives for a very long time. People have studied food colorings, they've studied emulsifiers. All of them seem to be bad. So, I think we still need more research into the mechanisms and repeated studies, but the body of data in this space is really quite compelling, which goes back to our dietary recommendations. When I say diversity, I don't mean entirely diversity. I don't mean including processed foods in that. And no one's really compared organic versus non-organic, but I think there's a general acceptance that eating quote unquote naturally is going to be best for our microbiome. And I don't think anyone has yet clearly defined what that is, but it's kind of common sense. What you might imagine as a healthy diet is going to be best for your microbiome.
As we start to get toward the end of things here, where do you see the work going in I don't know, 15 years?
My work or the field?
Well, I'm interested in what you're doing, to be honest with you, Suzanne.
I've had my lab for about 8 years now, so I still feel like we're in the very exciting phase of discovery. And so we will be continuing to do what we have been doing, which is diet studies and studying, disease pathology in patients. We're really interested in understanding this aspect of translocation, both transient and persistent. What does that mean biologically? And the body has some really amazing systems for controlling, the spread of microbes that go beyond the immune system. And so we're really trying to uncover that biology. So some of what we do is just fundamental curiosity and discovery. And not everything has to have a therapeutic end to it.
No.
But I do want to help people. I would love to see something we do end up as a therapeutic that could help people, whether it's a dietary recommendation or that becomes part of a treatment paradigm or an actual, medication, perhaps. That's always a nice thing to have as a goal. But we want really to think about, in terms of on the diet side, things that people can do to take control of their health at home, so they're not wholly reliant on the health system and taking matters into their own hands and trying to create optimal health. And maybe by understanding the microbiome, we might get a little closer to that. And so that's what we hope our contribution will be.
Thank you very much. It has been really wonderful chatting with you, Suzanne. I really appreciate your time. I think that listeners and viewers are going to just love the things that you've talked about today because it's relevant to everyday life. And I want to thank you for your time today.
Sure. Thank you for having me. It was a pleasure, Mark.
I hope that we can talk again soon. I've really missed our conversations.
Likewise. Hopefully at ASM.
Good. Best to you and yours.
Same.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes, with, as usual, fabulous links, can be found at microbe.tv/mm. If you like our work, consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Suzanne Devkota can be found in the Cedars-Sinai Division of Gastroenterology and the Cedars-Sinai Human Microbiome Research Institute in Los Angeles, California, a place I miss, honestly. Many, many thanks to David Renata, as usual, for fabulous editing, and Reber Clark for the quirky music. I hope you've enjoyed being part of our quality quorum for today. See you next week on Matters Microbial.